Hello everyone, it's Fiona again from the National Centre of Biotechnology Education. So today we are going to be discussing DNA technology. Now this is actually an area that is very relevant for the NCBE, since the NCBE was started back in the 1980s with the view to help educate students on the wonders of biotechnology and to develop resources to allow students to be able to study biotechnology in the classroom. So things like gel electrophoresis weren't available to do in schools before the NCB developed our little gel tanks that allow you to undertake gel electrophoresis using just carbon fibre as the electrodes and batteries as the power source. So this has made it easier to study DNA in schools and for you to gain practical experience in these important techniques. Okay, right, let's move on then. So today we are going to be talking about the polymerase chain reaction and other related technologies and their applications, such as paternity testing and forensics. So PCR is an amazing technique that allows us to amplify lengths of DNA. So you could start off with as little as one copy of a DNA strand that would be undetectable by standard methods. PCR then allows us to quickly amplify that DNA so that we end up with millions or even billions of copies that can easily be detected on a gel or can be sequenced. So this has allowed major breakthroughs in forensic science. So if a single hair or a small sample of a bodily fluid was found at a crime scene, then through PCR, any DNA that is extracted can be amplified and then analysed and compared to the DNA of any suspects. PCR is also used in paternity testing. So, how does PCR work? Well, we are basically trying to simulate what is happening in the body when DNA replicates, using the two antiparallel strands as a template for replication. So, let's talk first about what we would need in our PCR mix for the DNA to be amplified. We would obviously need our DNA sample that we're trying to amplify. We would then need what we call dNTPs. Now, these are the new nucleotides that can be added on to create the new DNA strands. We would need Taq polymerase, which, just like polymerase in our cells, adds on the new nucleotides and catalyses the condensation reactions needed to form the phosphodiester bonds that connect the sugar phosphate backbone of the DNA strand. We would also need primers, some water, some buffer that will be the correct pH for the Taq polymerase to work efficiently. And additionally, we usually use magnesium chloride as well, because this helps catalyse the phosphodiester bonds. So let's just go back then to primers. So what are primers? So these are specifically designed small lengths of DNA that are complementary to the DNA at the start and at the end of where you want to amplify. So these will bind onto the antiparallel strands and tell the Taq polymerase where to start adding on the nucleotides. So extension of the DNA then occurs in opposite directions. So once we have all our reagents needed for our PCR reaction, we can start amplifying the DNA. So there are three main steps to PCR. And they are denaturation, annealing, and extension. So firstly, just like when DNA replicates in our cells, the parallel strands need to separate in order for them to act as a template for replication. So in PCR, this is called the denaturation step, and it is done by heating the DNA to high temperatures. So this would be at about 94 to 98 degrees centigrade. And this causes the strands to separate. So next then, the primers need to anneal to the DNA. So this is our annealing step. So the primers obviously need to bind to the DNA so that the Taq polymerase can see where it needs to add on the new nucleotides. So this actually needs a lower temperature than the denaturation step, but the actual temperature for this varies depending on the composition of the primers. So depending on how many G's and C's you've got or how many A's and T's you've got in your primers will depend on the temperature that you need for your annealing step. But it's usually between 48 and 72 degrees. So once the primers then have annealed to the DNA, the PCR enters the extension phase. So this is then really the replication stage where the Taq polymerase comes along and starts adding on the new nucleotides. So this typically has a temperature of around 68 to 72 degrees, and this is optimal for then the Taq polymerase to start extending the DNA strands from the primers. So as the two original DNA strands are acting as templates, and one strand will be extended in one direction, and on the parallel strand it will be extending in the other direction. So these three main steps, so denaturation, annealing and extension, are just repeated over and over again, depending on how many cycles you want to run for your PCR. So a typical number of cycles is often about 35, but it can be more or less depending on how much DNA you need. So the DNA amplified in each cycle will then become a template in the next cycle, allowing the number of DNA strands to increase exponentially. So as you can see here, since we started with one double strand of DNA, by the end of the first cycle, this has become two double strands. And each new double strand contains an original DNA strand and a new strand, since DNA replicates semi-conservatively, which you would know if you've watched some of my previous DNA videos. So after the second cycle then, there are 4 double strands. There are 8 after cycle 3. There are 16 double strands after cycle 4, 32 after cycle 5, and you can see how you can end up then with millions of DNA strands after 20 cycles and billions of DNA strands after the 35 cycles. So with each step only lasting 1 to 3 minutes, a whole PCR program can be completed in a couple of hours, resulting in millions or billions of copies of DNA. So at the end of the PCR program, we usually hold the DNA at a steady temperature of between 68 and 72 degrees, so that any incomplete strands can be extended. So PCR is useful in all sorts of settings. So as I mentioned, it is extensively used in both forensic science and paternity testing. So for either, nowadays, the method that is commonly used involves variable repeating sequences. So these can be longer regions of 10 to 60 bases, or shorter regions of only 2 to 6 bases. So the longer regions are called variable number tandem repeats. The shorter repeat sequences are called short tandem repeats, or STRs, and the STRs are most commonly used now. So this method then, using the short tandem repeats, works by amplifying DNA by PCR at a selection of sites or loci, as they're more commonly called, that have short tandem repeat sequences. So different people will have different numbers of repeats at these locations. So for example, at a particular location, you may have 2, 3, 4, 5, 6 repeats of a particular short sequence. And of course, everyone has 2 alleles. So here you can see that this person at this particular site has 4 repeats on one allele and 3 on the other. So the 4 was inherited from the mother and the 3 was inherited from the father. Now then, if you analyse 10 to 20 of these regions, it gives you a number of repeats at each location. And this builds up what we call a genetic fingerprint or profile. So a person could have this as their profile for 12 different short tandem repeats. So the likelihood then of 2 individuals having the same genetic profile using this technique is vanishingly small. So it is a very powerful technique for identifying an individual in a forensic scenario or determining paternity. And because of its accuracy, it is now tends to be the gold standard, particularly for paternity testing. So this technique then is even more powerful if both the mother and the potential father are available for testing, since the short tandem repeat sequences that were inherited from the mother can then be accounted for and matched to her, making it then very clear which repeat sequences must have come from the father, and therefore should match if they are the biological parent. So here you can see in this example, so this child has inherited 4 short tandem repeat sequences at this location from the mother, and therefore the other allele that has 3 repeat sequences would have come from the father. So when multiple locations are looked at and found to all match one of the father's alleles, we can say with great confidence that this will be the biological parent. So you can see with this first child then that at this location anyway, this father could potentially be the biological parent. However, this other child, as you can see here, has 4 repeat sequences at this point, and the father does not, meaning that at this location they don't match, and therefore this cannot be the biological parent of this child. So for forensic analysis, if you have this sort of DNA profile for multiple suspects and a profile taken from some DNA extracted from the crime scene, then this can easily be matched to a suspect using this technique. So there are also older methods that can and sometimes are still used, both in paternity testing or in forensic science. So this technique uses restriction enzymes. So restriction enzymes then are enzymes that recognise a specific sequence of DNA and will cut at this site. So for example, you can see here that a common restriction enzyme will recognise this sequence highlighted in this box and cut at the site shown between the G and the A. So since then DNA varies between individuals with small mutations, etc. The number of cuts that a particular set of restriction enzymes would make would vary between individuals, and therefore the lengths of the resulting DNA fragments would also vary, giving a DNA fingerprint or profile that can be compared. So gel electrophoresis then is needed for this. So you can see here a potential genetic fingerprint for 4 individuals on a gel, with different length fragments at many of the positions, showing small differences in sequences. So let's now talk a little bit about a gel electrophoresis, which is another very useful technique that goes hand in hand with PCR for studying DNA. And many of you may have been lucky enough to do this yourselves in the classroom. So gel electrophoresis then allows the visualization of DNA on a gel and can allow you to estimate the size of DNA fragments. So first DNA would generally be cut up using restriction enzymes if it is a long stretch of DNA that is being analysed, so it can be seen more clearly on the gel. The DNA sample can then be loaded onto the agarose gel, and electrodes will be fitted either end of the gel so that an electrical current can be created. So DNA is negatively charged, so it will always move towards the positive electrode. So we therefore always put the positive electrode at the bottom of our gel tank so that the DNA will move down through the gel and towards the bottom of the gel. If you ever put your positive electrode at the top of your gel, you will lose your DNA quite quickly as it runs off the top of your gel. So you can see on my diagram here showing one of our NCB gel tanks then what an agarose gel looks like. So then depending on how big the DNA fragment is, will determine how quickly it will move through the gel. So smaller fragments will move much faster and therefore will travel further down the gel in a given time. Larger fragments will move much more slowly and therefore will travel less far. And this works on a gradient and can then be seen by looking at a DNA ladder, which is made up of DNA fragments of known size. So you can see a DNA ladder here. So fragments that are of an unknown size can then be measured against the DNA ladder, such as the bands on the gel here. So you can see that the ladder is in the first lane and then the DNA samples in the next 4 lanes. So the size of these bands then could then be estimated by measuring them across to the DNA ladder. So gel electrophoresis can be used in genetic profiling, but it isn't routinely done in testing labs since PCR methods are quicker and more reliable. But it is possible to look at DNA from different individuals on a gel after restriction digest and then see if the band sizes match. So for a suspect and DNA from a crime scene, or for a child and a parent in a paternity scenario. So you would expect then some of the bands would match one parent and some of the bands would match the other. The DNA, however, would still likely need to be amplified by PCR before it could be digested and run in a gel. So you can see a simple example of this taken here from our crime scene investigation kit. So you can see that there are 2 specific bands shown for the victim and 2 different bands that were produced from DNA that was extracted and digested that was found at the crime scene. Now then, we've got 3 suspects and we would expect one of those suspects to match these bands on a gel, showing that they were at the crime scene. Okay, so let's just recap then. So in this video, we have covered PCR, how this important method works, and what it is used for, so its broad range of applications. We have also looked a bit at other methods for analysing DNA, such as gel electrophoresis and using restriction enzymes to digest DNA. So tune in for our next video. Thank you.